I just want to say how honored I am and privileged I am to share the podium in this meeting with such fantastic prior speakers. It's a wonderful event, and I really thank Peter for putting it together. I'm going to be talking about, let me just say that what you've heard so far is an almost perfect introduction to where I'm going with this. We've gone all the way from the physics of dilute solutions to the physics of ordinary solutions, and then very concentrated solutions. And now I'm going to take this all the way to the ultimate extreme of, can we actually apply this to the most complicated system imaginable, which is an entire organ? And I think we'll show some evidence that this is probably possible to do. So the problem that I start out with in this talk, the problem of my life as a would-be organ banker, is that we have this wonderful solution that I think Brian referred to called M22. It's a 9.3 molar plus concentrated cocktail of cryoprotectants that's very mildly toxic to kidneys. It's very well tolerated by kidneys. It has ridiculously low critical cooling and warming rates. And we can warm rabbit kidneys by simple conduction means by 1 or 2 orders of magnitude higher rates than it should be required to vitrify and rewarm without any devitrification based on the properties of M22. And yet, we find that when we actually do the experiment, we do get a lot of ice formation in the kidney. And so you have to draw the conclusion that the cryoprotectant distribution into the kidney doesn't normally reach saturation, and therefore the system is more unstable than it should be. So the result of this is that I've got a problem banking kidneys, but in pursuit of that goal of banking kidneys, it creates a wonderful opportunity to really look at some very fundamental aspects of cryobiology and the physics of aqueous solutions as they exist in organized, complicated tissues in a vascularized organ. And I think we can learn a lot of interesting things from this. So you heard a lot of things that have been highly theoretical. I'm going to be the opposite. I'm going to be highly practical and kind of nuts and bolts. And I hope you'll forgive me if there's too much detail, but I think that the conclusion justifies all of the strain of going through all of this. Okay, so the reason that we can't distribute cryoprotectant well into organs is that the kidney is built by nature to thwart us from doing that. Essentially, the kidney gets 90% of its blood flow taken care of in the cortex only, and the outer medulla only receives 10% of the renal blood flow, and the inner medulla only receives about 1 or 2% of the total renal blood flow in normal circumstances. On top of that, the kidney vascular system in the medulla is arranged in countercurrent exchange mode, which also slows down the transport of cryoprotectants into the inner medulla. And this is all caused anatomically by bottlenecks in the renal vascular system, which are hard to overcome. As a result of this, if you look at what's going on in the urine coming out of the kidney, it lags a great deal behind the venous concentration, which of course lags a great deal behind the concentration of M22. And it's the concentration that we'd like to see in the urine and in the tissue itself. And it turns out the tissue is lagging even a little bit behind the urine in some cases. Now this would be a bit daunting, but there is a little bit of a ray of hope here because the kidney is a filter. And so in addition to having blood supply, or let's say the vascular system, as a pathway to deliver cryoprotectant into the medulla of the kidney, you also have glomerular filtration. So the blood normally gets filtered here at the glomerulus. The filtrate passes down this 1 cell layer thick structure known as the nephron. It comes back up to the cortex again, then it dives back down through the medulla a 3rd time before it gets delivered as urine. So in the course of all of that transport through the center of the kidney, there's opportunity for exchange of solute. So since filtration depends on pressure, if you increase the pressure, you can drive filtration. And considering that filtration could be a substantial fraction of total renal blood flow, in fact, a much higher fraction of total renal blood flow than the normal blood flow in the inner medulla, this is an attractive pathway to overcoming the equilibration problem. And this illustrates both the problem of not equilibrating and the possibility of equilibrating the kidney in view of these ideas. This is a kidney that was loaded with our standard cryoprotectant introduction procedure. We perfuse it at a mild, non-damaging pressure for 20 minutes. And when you cool it and warm it, we find that it forms about 6% ice under these circumstances. And to find out what that ice does to the kidney, we transplanted the kidney after vitrifying it and warming it up. And then after about 30 minutes, you take the kidney out, you flush it free of blood to the best of your ability. And what you see is that the vascular system in the center of the kidney, both the inner and outer medulla, have been destroyed, and you can't wash the blood back out of that structure. If you have a lot of ice in the inner medulla, it will actually propagate into the outer medulla, even if you didn't initially have ice there. So that's the problem that we need to solve. And here's proof that we can solve the problem, at least from a physical point of view. If we perfuse the kidney at 80 millimeters of mercury for a longer period of time, do the same experiment, there's no blood trapping in the medulla. So presumably this is mechanical damage from the formation of ice in this highly delicate structure, and that's been completely overcome. The problem with that is that this protocol is well tolerated by the kidney. This protocol will kill the kidney. So it's instructive, but this doesn't solve our problem. So we have to know what the minimum requirement is for avoiding medullary ice. And we have to know how much medullary ice might be tolerated. 6% ice is a lot of ice. Could a smaller amount of ice actually be okay? So part of the boundary conditions on this problem are answered by this experiment. So here's our normal protocol, perfusing M22 at minus 22, and by the way, that's why we call it M22. We perfuse it at minus 22 degrees Celsius to minimize toxicity. But if we ignore the toxicity to get an answer to the physics, and we perfuse at minus 3 degrees Celsius, you see the higher temperature allows a much higher flow rate to the kidney because the viscosity is dramatically reduced. And as a result of that, at the end of perfusion, instead of having the urine concentration being just about 91% of the full strength M22 concentration, you're up to around 96%. And if you now take biopsies of the kidney, you use a DSC to cool and warm them and check for ice, you find that with the standard protocol, you get your standard 4 to 7% ice in the inner medulla as a function of different warming rates. But in the minus 3 protocol, you get no ice. Something in the vicinity of 95% M22 is good enough. We don't have to get up all the way to 100% in order to successfully eliminate ice formation. And by the way, another point is that in the kidney, as far as we can measure, all ice that we see melting has been ice that formed during the devitrification process. So there's no ice on cooling. All ice is apparently formed on warming. All, meaning excluding teeny tiny nuclei, which is an infinitesimal volume fraction. I won't go into that. Okay, so that's part of the boundary condition on the problem, but we can't perfuse kidneys at minus 3 either because that would kill them too. But here's a case in which we could do something, and the kidney not only survived the perfusion process, it actually survived after being vitrified and rewarmed and transplanted. And so the trick here is to increase the perfusion pressure for limited time, just for 20 minutes, and then take the kidney out, vitrify it, warm it back up, put it back in the perfusion machine, drop the pressure back down to normal. This is the pressure going up here. This is the M22 perfusion phase. So your pressures increase during the M22 phase only, with step change up from 40 millimeters of mercury to 80 millimeters of mercury, then right back down when the kidney goes back in the perfusion machine. And the effect of that is that it lowered the amount of ice formed in the kidney based on theoretical observations on other kidneys from your standard 6% down to maybe 1.5%. So 1.5% ice formation in the inner medulla is compatible with survival of the entire kidney after vitrification and transplantation. But it's not good enough because the kidney was damaged. It supported life, but if you look at this curve, which is a postoperative renal function, this is creatinine level, which is a quick and easy way of measuring renal function. there's a little bit of injury, not too much. That's partly recovers, but it never settles down to a pre-treatment level of creatinine, so there's lingering injury. But it means we're close. So if we could just get the percent ice formation down below 1% somewhere, I think that we'd have an answer. So can we do that? Well, in order to approach that, we've developed a new pressure conditioning system recently that we call the flow lock protocol. With the old protocol, with introducing pressure very rapidly and dropping it very rapidly, it's a bit harsh, it's a bit arbitrary, and it's not very kidney specific because kidneys vary from kidney to kidney in how fast they perfuse. So we developed this protocol in which as the viscosity increases due to increased concentration of cryoprotectant and the decreased temperature, normally the kidney flow rate drops down. And if we left this with our normal protocol, the flow rates would go to very low levels right when we need to be perfusing the kidney the most in order to get the M22 in. But we prevent that flow from dropping down. So we lock in a flow rate of 0.2 milliliter per gram per minute. And then as a result of that, the pressure increases to various degrees depending on what the initial flow rate of the kidney was. And then as the pressure comes back down to normal, to 40 millimeters of mercury, then we release the we release the flow lock and allow the kidney to naturally return to a higher flow. But this experiment shows the extreme difference from kidney to kidney. So this kidney started off with a fairly pathetic flow rate compared to this one. As a result of that, the pressure went all the way up to 80, and we had to compromise a little bit on the flow until after the solution was diluted and the pressure came back down. So even though we try to impose the same protocol on all kidneys, there's a lot of biological factors to consider here too. In any case, if you look overall at what happens, this is the standard protocol, and what this is a total amount of urine passed through the kidney by the end of the M22 perfusion phase. Well, this is actually the whole time course. So the baseline method would put out about 0.4 milliliters per gram of kidney, but with the flow lock method for 20 minutes, it's about 0.6 milliliter per gram per kidney. So that's a 50% increase in the throughput of filtrate through the kidney. And if you lock for a longer period of time or you lock at a higher minimum flow rate, you can do a little bit better. Now, the consequences for the concentration in the urine are significant. So here's our baseline procedure coming up to close to 90% of full strength M22, and then we have the flow lock procedures going up to much higher and more promising levels. So what does this actually do to the vitrification tendency of different areas of the kidney? So this is results for cortex, outer medulla, and the urine actually when warming at 10 degrees a minute in the DSC. And you see that the amount of ice that we form is minimal in both urine and cortex, and very low above about 8.6 molar. The outer medulla forms much more ice. This is about 4% ice at the worst case. But again, we even see more than 2% ice at a much higher concentration. So we'd have to increase the concentration to 8.8 molar to take care of the outer medulla. Now, this is at 10 degrees a minute. That's what we would get if we took a kidney and just put it into a warm bath to warm it back up. But fortunately, Brian has recently given us a nice present. He's created this RF warming system that we've heard about earlier in the meeting, which is good for kidneys of all sizes, and has the miraculous property of giving us about the opportunity to go up maybe to 160 degrees per minute or something in that range, and then his system stops heating when you start getting up to dangerous temperatures for toxicity. So it's a very pretty system. So based on that, I would dare to consider what would happen if we could warm at 40 degrees a minute or even 80 degrees a minute. So if we look at what happens in these same kidney regions at 40 degrees a minute, you see zero ice in the urine, you see zero ice in the cortex, all the way down to 8.4 molar, which is a pretty dilute solution, one that the kidney has no trouble tolerating whatsoever. And the outer medulla is a little bit more recalcitrant. It requires somewhere between 8.5 and 8.7 molar, let's say 8.6 molar, to have devitrification suppressed in it, but that's a lot better than the 8.8 molar we saw before. But what about the inner medulla? Well, the inner medulla is a lot worse. So here's the 8.6 molar limit for the outer medulla, above which no ice would form essentially. And for the inner medulla, you have to go up to that 8.8 molar level in the urine in order to suppress it. But it turns out that this picture is far more interesting than it looks. These are all means of 2 biopsy samples per kidney. So we took the kidney, we cut it in half, we took a cortex sample and an outer medulla sample and an inner medulla sample from both halves of the kidney, and they're not matched up. So if you look at what happened in the individual biopsies, you see a remarkable thing. Instead of just having kind of a smooth approach down here to 0 ice, you see this fantastic dichotomy between the 2 different biopsy samples in many experiments. And in the lucky biopsies, the inner medulla behaves just like the other parts of the kidney, and you can tolerate, you can escape ice formation at relatively low concentrations, even approaching that 8.4 molar level that we saw in the other tissues. The problem is that the biopsies are not consistent with each other. You can suppress this heterogeneity by brute force, just by perfusing more CPA into the kidney. You can snuff this off in a very narrow concentration region. But the question is, where is this all coming from? By the way, before we go there, I just have to say that this heterogeneity of the inner medulla was observed in that surviving kidney, and is probably the reason why the kidney didn't function very well. It turned out that half of the kidney had normal surviving histology after we took this kidney out of the animal 48 days after we transplanted it, but the other half of the kidney had dead inner medulla. So this problem has persisted over a long period of time. So if you think back to all of that tremendous heterogeneity from biopsy to biopsy, what would happen if you now compare the amount of ice formed in the biopsy sample against the TM of that biopsy sample? And if you do that, you find that there is no disagreement between the samples at all in that regard. You have a universal relationship between the amount of ice forms and the melting point of the system. Whether it's inner medulla, outer medulla, or cortex, everything behaves the same way. And by the way, this box here, in which you see no ice formation on warming of the inner medulla, that takes place at TMs between -40 and -50, which on a theoretical basis is what you need to vitrify stably, because if you have a TH below you have a TM below -40, your TH will be depressed to below TG. Okay, so using a phase diagram of dilutions of M22, we can convert the melting point differences in the melting points in the concentrations, and we find in that way that as long as your tissue, whatever it is, has a local tissue concentration greater than about 8.6 molar, it's stable. It's not going to form any ice. If it's less than that, you get into some trouble. So there's a distinction between tissue concentration now and urine concentration that I told you about before. And if you look at the amount of ice that formed as a function of the mean concentration of the biopsy pairs, you look at the difference in the amount of ice that formed between the biopsy pairs against the mean concentration of the biopsy pairs, you find a kind of a rational relationship between those variables. Again, snuffed out when you get to about 8.6 molar tissue concentration. And now, if you translate the ice data into concentrations, you see basically the same effect. And so now we can plot those two things against each other to find out how much of a difference in concentration there is from region to region within an inner medulla that's driving all of that ice formation. And the results are fairly extraordinary, that as long as you're below a concentration difference from biopsy to biopsy of about 0.2 molar, there's very little chance you're going to see significant ice. But if you may see significant ice if you are somewhere between 0.2 and 0.6 molar in your concentration differences from region to region within the kidney. Now, for perspective, that's about 2% to 6% of the perfusing concentration. It's still a fairly uniform concentration, but it's still, the differences are driving the whole problem. Okay, so one other point on the physics, and then we'll go to the biology. In view of the fact that we might be able to heat fairly rapidly, how does the amount of ice formed at higher rates compare to the amount of ice that would form at a warming rate of 10 degrees a minute? And so there's a concentration dependence of that. So this is for the ratio of ice at 40 to ice at 10. And you see, as long as you're fairly dilute, like below 8 molar or something, warming rate doesn't really help you that much. But as you get higher in concentration, you can snuff out ice formation fairly effectively by just increasing the warming rate from 10 to 40 degrees a minute. And the results are more dramatic if you increase from 10 to 80 degrees a minute. You snuff out ice formation at local tissue concentrations of about 8.3 molar, which is remarkably dilute. So that higher warming rate is very attractive. Okay, this is actually going to work, I think. I hope. Okay, so now let's look at the biology. We have a fairly good idea what we need to do, what happens when we try to do it. So if you transplant kidneys after different urine concentrations have been achieved, there's a general tendency, unfortunately, for damage to increase as the saturation of the kidney with cryoprotectant increases, which is not too surprising. But you need to look at this in slightly different ways. First of all, look at those guys. They were out here at the highest concentrations ever, and they had some of the very most acceptable creatinine peaks that there were. And also, this is the dividing line for being okay to warm at 40 degrees a minute. You still have some good guys out here. You do have some problems, but if we could go above 40 and go between 40 and 80, you're in a fairly pleasant zone here in which the kidney will always survive. And if we can look at the probability of recovering normal function independently of the peak concentration, because the peak just tells you how much damage there was, it doesn't tell you if that's reversible. If it's reversible, you don't really have to care how much damage there was. And so 1 here means that all damage was reversible. In other words, we could return a normal creatinine level, and 0 means you couldn't. So by the standard of warming at 40 degrees a minute, you do have some losses above that standard, although you do have some good survivals. But you have essentially no losses in the region between the criterion for 40 degrees and 80 degrees a minute, which we think we should easily be able to achieve. If you look at the details, these are kidneys that had the magic urine concentration greater than 8.83 molar cryoprotectant. So these guys are immune to devitrification as long as we can warm them at least at 40 degrees a minute. And you see, yeah, we do have some that have high creatinins, but they ultimately recovered to below a creatinine of 2, so they recovered complete normal function. And we had one guy that was phenomenally able to tolerate these very stabilizing concentrations of cryoprotectant. And we had this one other guy out here that's extraordinary because its creatinine, its concentration was about 9 molar, way in excess of what you need to have stable vitrification, and it was just fine. It just didn't mind at all. So, and then if you look at the trend here of peak creatinine level against the concentration in the urine that you actually achieve, it's essentially a horizontal line. So we actually don't think that there's a necessary problem with cryoprotectant toxicity here. There's also biological variability from kidney to kidney. There's a lot of different techniques that we use to do these experiments. All of that may be important. The pressure exposure may be important. So it's fairly optimistic if you think about it that way. And then if you look at the kidneys that we think would be immune to devitrification if warmed between 40 and 80, this is a perfectly normal creatinine curve, very acceptable. This is basically the same level of damage we see with our standard M22 protocol, which is nowhere near equilibrating enough. But these guys would be equilibrated enough, and they have no problem with it. There's also, again, no trend for increased damage with increased concentration. So what do we conclude from all of this? we think that the main obstacle to cryopreserving the kidney is devitrification of the inner medulla. We can measure the stability of the kidney by following the urine concentration, which is a non-invasive thing that you can do in real time. At 40 degrees a minute, the critical concentrations that you need in the urine go from about 8.4 molar for the cortex to 8.6 molar. For the outer medulla, and 8.8 molar. For the inner medulla, very narrow concentration differences, having profound differences in terms of the amount of ice that's observed. And the main problem with the inner medulla seems to be relatively small differences in concentration from place to place. But that higher concentration, if necessary, can be tolerated, and some kidneys do very well with it. But if we could warm at 80 degrees a minute or even less, we should be able to lower the critical urine concentration to a level that all kidneys will tolerate easily. So based on all of that, this looks pretty exciting. And so unless some other problem arises that we're not quite aware of yet, in principle, we may actually now be at the point where once Brian is done putting the finishing touches on the electromagnetic warming system, we may be able to routinely vitrify kidneys and warm them up and have them survive as long as we can do the warming. And we hope to find out the answer to that question by the end of the year. Wish I could have told you now, but we're not quite there yet. And thanks for the patience. I wanted to acknowledge the surgical team and the tissue analyst that did all of the DSC measurements for us. And these guys also put together a great number of the slides that I showed you today. I'll take any questions if there's time. Thanks.
I've known Greg a long time. He's got tremendous courage over these years of very, very difficult problems. So, yeah, Dr. Humphrey.
What are the issues of scaling this up into the outer standpoint of human physics for all your products?
That's kind of unknown. Generally speaking, what we observe with rabbit kidneys is that bigger kidneys have more vascular resistance, so they're a little bit harder to perfuse. But I don't know that that really translates between species. So I think the main issue is likely to be the electromagnetic warming side of that. But Brian assures me that the penetration depth of RF energy is such that we should be able to handle human kidneys almost as well as rabbit kidneys. So I guess the other question is one that I can't address at all, and that's species differences. Maybe the human kidneys are more susceptible or whatever, but the normal rule of thumb would be that humans are tougher than other animals. You know, we live to 80, rabbits live to 8. So we tend to be made of sterner stuff. We tend to be able to survive these kinds of things better. So I think that's actually in our favor. We actually have a room at 21st Century Medicine that is isolated from all the other rooms. We can receive human tissues for study. We have received human organs for study in the past, and if we get there, we will actually be able to perfuse human organs with vitrification solutions and test them in vitro before we propose any clinical trials. One other point is that in terms of chilling injury and susceptibility to cryoprotectants, I was able many years ago to make a direct comparison between rabbit kidneys and humans and found no difference. So that's N is equal to 1, but it's reassuring.
Let me just pose one. There is an alternative system to avoiding isolation in tissues, and that's one particularly developed by Fahy called liquid cooling, where you follow the Fahy diagram, progressively increasing concentration, so that you end up getting no external life forms at all. He's used that for cartilage, and I think the algorithm as well. Do you want to make? I don't want to make.
Sure. Yeah, sure. I was inspired ultimately at the root of all of this by John Ferrant, who came up with that idea a long time ago, 1965. It was a phenomenal concept and result that he published in Nature, showing he could take a whole organ, guinea pig uterus, down to dry ice temperature and back and have it recover fully because he avoided all ice formation in the system. But he had to use multiple steps of DMSO. He used DMSO, which is too toxic to just use outright. So he put in a little bit, but that would depress the freezing point. And at the lower temperature of that freezing point, the toxicity was reduced, so we could add more DMSO. So you just repeat that all the way down to dry ice and back, and he had complete success. And he proposed at the end of that this would be a way of preserving things for which you need 100% cellular survival. And I thought, what a concept. But the problem with that is that if you want to do liquidus tracking, you have to be able to track the liquidus. And that's impossible in a perfusion situation. It's hard enough to perfuse kidneys at -22, as you saw. And I showed you the difference between perfusing at -3 and -22. So we're forced to cram all that CPA in at very high temperatures and do everything possible to control the toxicity so we can get away with that. And so far, it looks like that's working. So I think that's the way to go for us.
I was just curious, Greg. Nice progress. It's really exciting. Do you think the differences you're seeing in your samples in terms of ice formation in different parts of the tissue have anything to do with differential permeabilities or diffusibilities of the different components in your vitrification solutions?
It could be. It could be. We've actually looked at that. All of those urine concentrations that I gave you make the rash assumption that the filtrate is an exact duplicate of the perfusate, just less concentrated.
Okay.
But we do find that there are slight concentration differences from CPA to CPA compared to what's going in. So there's differential distribution into the urine. We don't know if that's due to differential filtration or if that's due to differential absorption, and if so, where the differences are coming from. We don't really quite know that yet. But we could determine that because we have an HPLC method that can analyze tissues. And we have extensive data. I just haven't really completed the analysis of that yet. But it's a great question.
Okay, thanks.